Hello everyone. Today I will show you how to retrieve pathway IDs and names for a gene using Python. So let's get started. First of all, we have to import necessary libraries. In order to send a request to the KEGG API, we have to import or to install a library known as request. So we can install this library using pip install request. If this library is already installed on your computer, you can use import requests. I have this library already installed, so I will run import requests. Secondly, the second step is to define the function. So in order to define a function, first of all we have to write a def keyword, then the name of the function. The name of the function that we want to write is get pathways. And here we want to give one input argument that is known as gene symbol, for which we want to find KEGG pathways. Then we will use the URL to convert our gene symbol into the KEGG ID. So we can did it like this: f string, then we have http :// rest.kegg.jp. Then now in order to find the KEGG ID against our gene symbol, we will use find/gene API endpoint. Find/gene API endpoint, and for which symbol we want to find out, we'll write exactly gene_symbol variable. So whatever gene symbol will give to this function, in this URL will find the IDs against that gene symbol. Then we will store this in a variable known as gene response so that we can access it easily. So this URL will now basically find the KEGG ID and will store it within the gene response. Now we will use the request library to get information against this URL. Now this URL is basically not sending request. We will send a request with the help of the request library. So this request library will basically use this URL and will write the gene symbol that we will provide to this function, and we will call it. For example, this gene symbol can be TF gene symbol or BRCA1, etc. So this request library will use the .get function to get the information from the KEGG API KEGG database, and will use this gene response variable. So in order to easily fetch the information, and we will store it in a variable. Here we can write it like gene search URL, and we'll store it in a gene_response, and here it is gene search_URL. Now basically let me explain again like how it works. First of all, we will write this URL. This URL will basically send a request. This URL will be basically used by the request library to get the KEGG ID against this gene symbol, and we are storing basically this URL within a variable known as gene_search_URL. Then in order to get information against this gene symbol, we will use the request library. We'll use this request library and we'll use its function .get to fetch information against this gene symbol, and we'll store this information, this output, with another variable known as gene_response. So now this gene_response will will show results consisting of different methods and attributes. This gene underscore response will consist of dot text. It will contain dot text information. It will contain dot status underscore code information. It will contain dot JSON information and dot content. But here we only want to print gene underscore response dot text because we doesn't have any concern with other parts, so we only need this main body of our gene response. So we can print this gene response and we can get it. So let's try it, either this function is working or not. Get underscore pathways. Now this is the way to call a function. First of all, we'll write the name of the function and then the argument that we want to provide it, BRCA1. Okay, it's working exactly and how we want. So now, in order to check that either the information that we want to find against our gene symbol is present in KEGG database or not, we will check it with the help of dot status code. So first of all, we will write if statement, if gene underscore response dot status code is not equal to 200 or not gene underscore response dot text dot strip. We will print an F string, no gene info is present for gene symbol and then we'll return it. So how we are basically checking the status code. So if the request is successful and our request gets successful and it has reached the KEGG database, so its status code is equal to 200. This means that our result has been successfully fetched out from the KEGG database. But if the status code is not equal to 200, this means that our information that the information that we want to find from the KEGG database have not been fetched out, or the second condition can be that that information has been fetched out but it is not valid. So in order to check the validity of our data, that either the information that we are fetching out and storing it within the gene response variable is either valid or not, so we are going to use not gene underscore response dot text dot strip. Now here basically I have already told you that we will work with only with the gene underscore response dot text attribute in order to work with the text body, and strip will basically remove the starting and the ending byte spaces. So if either of these conditions gets true, it will shows that no gene information is present for this gene symbol, for example BRCA1, that stands for breast cancer 1, and will return nothing. And here our function will stop working and will move to the end of the end line of the code. Now if this status code is equal to 200 and information is successfully fetched out from the database, then we will store it in a variable. Then we'll store this information of this ID with another variable like gene underscore response dot text. First of all, we will split this text part with the help of new line, and with the help of a new, this is basically a new line that we want to split this text body with new line, and we want to store its zeroth position values into a variable known as gene data. This gene data will basically contain the information that we want to store in a variable, and then we will use this gene underscore data dot split. Then we'll basically split this line further by tab, and will store 0th position into the KEGG gene ID. And we can print this KEGG gene ID KEGG gene ID. So how this response is basically working and how this tab is basically working. So for example, it will give us information like HSA 672 \t. This means that this part will contain tab BRCA1 breast cancer 1. So this is the basically main line from which we want to fetch this ID. So this is basically, this shows the ID for our gene symbol, and this is the full name of our gene. And so here HSA represents that this gene belongs to Homo sapiens, and this is the unique identifier that is assigned to this gene symbol in order to differentiate it from other genes belonging to same species or the other species. So here we will separate it from tab and store the 0th position value, that is HSA 672, within the KEGG gene ID variable, and we can print it by using f-string f quotes KEGG gene ID KEGG gene ID variable, and we'll stop it like this. So now next step is to find the pathways that are present against this KEGG ID. So basically what we have done so far is, first of all, we have find out the KEGG IDs against our gene symbol. Now why there is need to find the KEGG ID against our gene symbol? Because this KEGG ID will basically help us to find KEGG pathways against that ID. So it will give us KEGG pathways against it. That's why we are finding the KEGG ID. So our next step is to find pathways linked to the gene that for which we are working for. So first of all, its process is same as like we have sending request to the KEGG database to find out the KEGG ID. It will be like this: http://rest.kegg.jp, and here the endpoint that we are going to use is link/pathway/KEGG gene ID. This is basically the variable that is containing the KEGG gene ID for our gene symbol, and we are storing this URL in a pathway underscore URL. Then we will send request with the help of requests library pathway underscore URL, and we'll store this response in a variable known as pathway underscore response. So now in order to check that either this request is successful or not, we'll use the same method of dot status code if pathway underscore response dot status code is not equal to 200 or not, pathway underscore response dot text dot strip print f string like no pathways found for gene symbol gene underscore symbol. And will return none against it. Then we have pathway underscore response dot text dot strip pathway underscore response dot text dot strip dot split \n. So here basically we are going to basically this text part into the we are removing. I have already told you that pathway underscore response dot txt is basically for which we are working for, and we'll split it with the help of a \n, and we'll store this data into a variable known as pathway underscore data. Now this pathway underscore data will basically contain pathway underscore. So for those pathways we are only storing the IDs within a list. We will initialize an empty list, and then we will use a loop like for line in pathway underscore data. We will pathway underscore IDs dot append line dot split it with the tab, and then split it on the basis of first position, and will store this value in. So here what we are doing here, we have initialized an empty list, and then we are using a loop on this pathways data information, and we want to basically store only pathway IDs within this list. So we will run line by line we will go through line by line with the help of a for loop on pathways data, and then we will append this line by splitting it by tab, and will store its one position. Means as we know that in string indexing starts from 0, so we will skip the first 0th part and will store only the first part of this string within and will append it within a list known as pathway IDs variable that is stored in the pathway IDs variable. Now here all of the IDs will get stored within this list. And now in order to get pathway names also, we will use the same mechanism. First of all, we will initialize an empty list, and we'll use a loop for it for pathway underscore ID in pathway underscore IDs. Now here we'll basically use each ID from this list to get the name against that pathway. It will look like this. For example, if the ID of any path is this, so the name that can be stored for it is like glycolysis. It will work like this. Okay, so it will use this pathway IDs to get its pathway name, that can be, for example, glycolysis. So for pathway ID, in pathway IDs, we will send the request again to HTTP with the help of, so here we will use the same mechanism, that is http://rest.keg.jp. Here we are using the endpoint to get the GEG names is get/pathway, and we'll use pathway ID, and we'll store this variable in pathway underscore info underscore URL. Then we will send requests dot get by using this variable, and we will store this pathway underscore info underscore response. And now we will check the status code that either the name is available against this ID or not. We'll use this if pathway underscore info dot response dot status code is not equal to 200, we will use this same pathway underscore info underscore response dot text dot split, then we are storing only its first part within it, and then we are splitting it again on the basis of name. And then we are using its first position, means so in string basically indexing starts from 0, so here we are skipping 0th position, and so we're working on first position, and then we'll split this first position again on the basis of its first position, and we'll remove the white spaces from it, and we will store this information into a variable known as pathway name. Here we will append these pathway names dot append, will append these pathway IDs and pathway names into this list like this: pathway underscore ID and pathway underscore name. First of all, its ID will be stored, and then its name will be stored out in this list. Now we want to print these pathways. We'll use f string and we'll say pathways for gene underscore symbol, and here we will use loop on it in order to print names of pathways and IDs. From this list we will use two variables, pathway IDs and pathway name. In pathway underscore names list we will print them separately like this, like pathway IDs will be like PID and pathway name will be like P name. And here we will return pathway underscore names. So here basically we are using this pathway underscore names list, and we are using for loop in order to print each value within this list by splitting them on the basis of their IDs and their pathway names. Now we will call the function that I have already told you that first of all we will write the name of the function, and here we will give the gene symbol for which we want to find the pathways IDs and pathway names. So the pathway gene symbol that we are going to provide is BRC1, and so let's here our code get here we are basically calling the function, and let's see that what is the output. We can basically run this cell from here, and we can also press shift plus enter in order to run the code. So here our code is running to show you like this and this. It will take some time to find the IDs and names, so be patient with it and do not close your window. Excellent, our result has been fetched out from the KEGG database, and it is storing basically the IDs like this and names like this. So this is basically the ID of our pathway, that is HSA01524, and its name is platinum drug resistance, and so basically our gene, that is BRCA1, is involved in these pathways that are 1 2 3 4 5 6 7. So this gene is basically involved in 7 pathways within Homo sapiens species. And that's it about this video. In this tutorial we used Python to interact with KEGG database and to retrieve pathways information for a gene sample. Thank you so much for watching. If this was helpful, don't forget to like, share and subscribe. See you in the next video.